So my name is Iselle Barrios. I was in the rotor lab this summer, and my project is about looking at the differences in cellular growth patterns between rough and smooth Arabidopsis organs. So robustness in biology is defined as the ability of an organism to grow organs that are of the same shape and size, even despite environmental variation. If you think about it, we see robustness all around us. Trees, like this mesquite tree here, grow thousands of leaves throughout their lifetime, all of which have roughly the same size and shape. Animals, like this giraffe here, are able to grow limbs that are consistently of the same length. Robustness is even something that we see in humans. Human arm lengths tend to match very closely. However, even though robustness is such a universal property of living things, it's something that we still don't understand very well. So our lab studies Arabidopsis to understand the mechanisms that underlie robust growth. So what you see on the left here is a wild-type Landsberg Arabidopsis inflorescence. Essentially, it's just a bunch of different flower buds at different stages of growth. Each of those flower buds is surrounded by 4 green leaf-like organs, which are called sepals. On the wild-type plants, these sepals are relatively smooth, and within each flower bud, all 4 sepals are of the same size. Essentially, they exhibit robust growth. Now, our lab was interested in finding a mutant that doesn't exhibit robust sepal growth. During a mutant screen, our lab found 3 variable organ size and shape, or VOS mutants. On the right here, what you see is a VOS3 inflorescence. As you can see, it has all these weird outgrowths and creases in it that make it look very different from the wild type. Additionally, these outgrowths and creases are located seemingly randomly throughout the sepal. In other words, they exhibit a loss of robustness in growth. So these differences are just as dramatic when you look at the sepals up close. This on the left here, you can see a wild type Landsberg sepal, and on the right, you can see a VOS3 sepal. While the wild type sepal is relatively smooth, again, you can see on the VOS3 sepal that it's covered in all those lumps and creases. So you might be wondering, what exactly causes this lumpy sepal phenotype in VOS3? Well, our lab found that this was due to a single nucleotide change in the promoter of the gene asymmetric leaves 2, which confers gain of function upon AS2. So in wild-type plants, AS2 is expressed only on the inner side of the sepal. However, in the VOS3 mutant, AS2 is expressed on both the inner and the outer side, which is what leads to this lumpy sepal phenotype. So we were interested in looking at the differences in cellular growth patterns between the wild-type sepals and the VOS3 sepals. To do that, my mentor, Dr. Avinash Singh Yadav, live imaged growing sepals of wild-type and VOS3 flowers every 24 hours for 6 days. So this is the day 1 image of a wild-type sepal. You can see the emergent sepal right here, and in the background, that kind of curvy thing, that's the floral primordia, which is where all of the floral organs grow out of. By day 2, the sepal has grown to cover the floral primordia, which it does primarily to protect it. By day 3 and 4, you can start to see the formation of structures such as a trichome, which is a protective structure, and you can start to see the formation of giant cells, which are, as their name suggests, just cells that are much larger than the others. By the time we get to day 6, you can see the sepal's really grown a lot. And there's one important thing that I'd like to draw your attention to. Despite the giant cells, which kind of have a tendency to bulge out from the rest of the sepal, the surface of the sepal is very smooth. Now, we see a very similar pattern when we look at our second replicate of the wild type. You start off with a small bud that has the floral primordia exposed. The sepal grows to cover that. And by the time we get to day 6, we have a much larger flower bud that has, despite giant cells, a relatively smooth surface. Now, when we look at the VOS3 mutant, we see a very different pattern. For the first couple days, it looks more or less normal. It's a lot like the wild type, relatively small, relatively smooth sepal. When we look at days 3 and 4, you kind of start to see the formation of structures that we don't see in the wild type. So for example, you have something that looks like it's becoming an outgrowth here and a crease there. These only get more dramatic when we look at days 5 and 6. You can see by day 6, you have all these crazy outgrowths on the side, and there's that crease there, and it's just a mess. So we see something very similar when we look at our second replicate of VOS3, excuse me. Again, we start out with a relatively normal looking sepal that gradually gets more lumps and creases as it grows. Now, there's one important difference that I want to point out. The location and shape of these lumps and creases on the second VOS3 replicate is different than it was in the first VOS3 replicate. Again, this suggests to us that the VOS3 sepal shows a lack of robustness in growth. So as I mentioned, we were interested in looking at differences in cellular growth parameters between the wild type and the VOS3 sepals. To do that, we use morphographics image processing software to extract data from the images that I showed you earlier. The first cellular growth parameter that we looked at was area growth rate. What you see here is a heat map of area growth rate for the first 3 days of our wild type first replicate. As you can, cells that grow faster are in red, and those that grow slower are in blue. As you can see on day 2, there's relatively fast growth throughout most of the sepal. By day 3, that growth has slowed somewhat, and the cells that are still growing very quickly are concentrated around the edge of the sepal in a uniform pattern. Now, VOS3 is quite different. Oh, yeah, okay, there it is. VOS3 is quite different. On day 2, there's less fast growth overall than we see in the wild type. And on day 3, the distribution of quickly growing cells is very different. They're scattered seemingly randomly throughout the sepal and not in the nice uniform pattern that we see in wild type. Now, if we look at days 4, 5, and 6, we can see that wild type displays kind of a shifting fast growth front, where the cells that are dividing quickly kind of move down toward the base of the sepal. We don't see this kind of a pattern in VOS3. Again, in days 4 and 5, the quickly growing cells remain seemingly randomly distributed throughout the sepal. And by day 6, there's very little growth at all. Now, when we look at our second set of replicates for wild type and VOS3, we see again similar patterns. You start off with that very fast growth on day 2, slower growth on day 3 with a uniform distribution around the edges of the sepal. When we look at VOS3, we again see it looks like slightly less fast growth on day 2. And on day 3, you again have that patchy random distribution of quickly growing cells throughout the sepal. When we look at days 4, 5, and 6 for wild type, again, we see that shifting fast growth front down toward the base of the sepal. Whereas when you look at VOS3 days 4, 5, and 6, again, there's kind of a patchy random distribution of quickly growing cells, and there's less fast growth overall in VOS3 than there is in wild type. So after looking at differences in cellular growth rate between the VOS3 and wild type sepals, we decided to look at differences in cellular growth direction to see if perhaps these could explain the lumpy sepal phenotype that we see in VOS3. So what you see here is a heat map of anisotropy for the first 3 days of wild type replicate 1. Now, anisotropy is essentially just a measurement of how directionally a cell is growing. So if a cell is growing very quickly in one direction and not very quickly in the other, then it's growing very anisotropically. Cells in this graph, cells that are growing very anisotropically are in red, while those that are growing more symmetrically are in blue. The white lines on each cell represent the direction in which the cell is growing the fastest. So you can see that on day 2, we have a lot of anisotropic growth, and most of that is aligned upward as the sepal grows to cover the floral primordia. By day 3, there's much less anisotropic growth. And what anisotropic growth there is along the edges of the sepal. There's almost none in the center. When we look at VOS3, we see something different. There's less anisotropic growth overall on day 2. And when you look at the directions of growth on VOS3, it's more sideways than it is the wild type, which is very well aligned upward. When we look on day 3, the distribution of anisotropically growing cells in VOS3 is kind of random, sort of like we saw with the area growth rate. It's not uniform like the wild type is. When we look at days 4, 5, and 6 in wild type, the anisotropically growing cells shift from the edges of the sepal down toward the base. When we look at VOS3, however, again, you see kind of a random patchy distribution of those anisotropically growing cells, and there seems to be less anisotropic growth overall. So now that we looked at the general differences in anisotropy between VOS3 and wild type, we decided to kind of zoom in on the VOS3 sepals to see if perhaps the locations of anisotropic growth could give us some insight into the formation of lumps and creases. So what I've done here is highlighted with arrows areas that will become lumps and creases in the later days for these VOS3 sepals. As you can see, the areas that later become lumps and creases, I think that one later becomes an outgrowth, this later becomes a crease. They show relatively anisotropic growth on day 2, and also to some extent on day 3. Now, this is a really exciting finding because to us, it suggests that anisotropic growth precedes the formation of outgrowths and creases in the VOS3 sepal. When we look at days 4, 5, and 6 for VOS3 sepals, we see a slightly different pattern. I've again highlighted the same areas with arrows. Now, what you see here is that areas that have outgrowths and creases tend to show anisotropic growth around them. For example, if you look at this crease here, there's a lot of anisotropic growth in there. or this outgrowth here, some of the cells around the outgrowth are growing anisotropically. So this suggests to us that there's some connection between the existence of an outgrowth or a crease and anisotropic growth. So when we look at our second set of replicates, we again see a similar pattern. Oh, my bad. On day 2, you see a lot of that anisotropic growth in wild type, all aligned upward as the sepal grows to cover the floral primordia. And on day 3, you see the anisotropic growth uniformly distributed around the edges of the sepal with none in the center. Looking at VOS3, again, you see that the directions of growth on day 2 are more sideways than they are in the wild type. And when you look at day 3, the distribution of anisotropically growing cells is much more scattered and patchy than it is in wild type. Looking at days 4, 5, and 6 for wild type, you again see that the anisotropically growing cells shift from the edges of the sepal down toward the base. In VOS3, however, again, we see that kind of a random patchy distribution of anisotropically growing cells, and overall, there seems to be less anisotropic growth in VOS3 than in wild type. So we did this same kind of thing we did with our previous VOS3 replicate, where we looked at areas that will later become lumps or creases on their earlier days. Again, the areas that later become creases, I think that one's an outgrowth, these two are creases, they show anisotropic growth in day 2 and day 3, which confirms our suspicions from earlier that many of the creases and outgrowths in VOS3 are preceded by anisotropic growth. Looking at days 4, 5, and 6, I've again highlighted the same creases and outgrowths. And again, to some extent, we see the existence of anisotropic growth around areas where there's outgrowths and creases are. For example, this outgrowth here, or that outgrowth there, some of the cells around it are growing anisotropically. So general conclusions we can draw from this, that in early days, VOS growth exhibits more sideways growth than wild type, and that in both samples we looked at, anisotropic growth precedes the formation of those outgrowths and creases in VOS3. So the final growth parameter that we looked at was cell division rate, or the number of times that a cell had divided within 24 hours. What you see here are heat maps of wild type and VOS3 cell division rates, where cells that have divided more times are in red, and those that have divided less are in blue. Now, what we see here is that there seems to be a lot more cell division in the VOS3 sepals in general than there is in the wild type. This pattern changes when we look at days 4, 5, and 6. Wild type shows kind of a cell division front, whereas the distribution of cells that are dividing rapidly shift down toward the base. VOS3, however, shows more of a patchy random distribution of quickly dividing cells, and there seems to be less cell division overall. Oh, shoot, wrong button, especially in days 5 and 6. When we look at our second replicate of VOS3 and wild type, we see very similar patterns. Again, especially in day 3, there seems to be more cell division overall in the VOS3 sepals than there are in the wild type sepals. When we look at days 4, 5, and 6, again, there's that shifting cell division front in the wild type as the distribution of quickly dividing cells moves down toward the base. Whereas in VOS3, we see a much more random distribution of quickly dividing cells. And I'm really bad at this clicker. And especially on day 6, we see much less fast cell division. So general conclusions we can draw from this. So because of the association we saw between the formation of lumps and creases and anisotropic growth, VOS3 proves to be a very interesting mutant that we can use to study the connections between cellular growth direction and robustness. In terms of area growth rate, wild type is sepals are able to maintain some amount of fast growth throughout the 6 days that we image them for, whereas the VOS3 fast growth dies away sooner. We can say that outgrowths in VOS3 are preceded by that anisotropic growth. And finally, with regards to cell division, while VOS3 initially seems to divide more than wild type, that cell division dies away more quickly. So I'd like to thank my mentor, Dr. Avalash Singh Yadav, and my PI, Dr. Adrian Roeder, and everyone in the Roeder Lab just for being so supportive and welcoming this summer. It's been a really great experience and a really great environment. And of course, I'd like to thank BTI and Cornell for hosting this program, and the NSF, USDA, and NIH for providing funding. And of course, all of you for listening.